Okay.
I can do that, right?
Yeah. So what I do, number one, is we look at neglected tropical diseases. You know, these are diseases that are they are basically inherent in Africa, and we use nuclear magnetic resonance and micromolecular spectroscopy to solve the structure. We do drug discovery design for diseases such as schistosomiasis, which I'll be talking on today, diabetes, TB, cancer, and just recently we started looking into monkeypox. In fact, we actually have a proposal which we've just finished, which we're going to submit to NRF for funding into monkeypox drugs. Also, we also looked at the fabrication of point-of-care diagnostics for rapid testing, and we do protein-protein and protein-ligand interaction studies using biophysics methods. And then we started working with our colleagues in chemistry, looking at nanobiotechnology, whereby we can develop nanoparticles by which we can encapsulate drug molecules and put a targeting molecule on it, which can then be targeted straight into the brain, because our own idea is to actually target glioblastomas, that is cancer of the brain. And lastly, I also do a bit of mathematical modeling where we model disease episode and how much it will cost government to spend, because oftentimes when you speak to government, they looking for numbers. They don't want all the sciences that we talk about. They just want you to tell them the number, how much are they going to spend, and how much will it cost government, will you take from the fiscus. So we do a bit of mathematical modeling where we model climatic changes in and their influence on the spread of infectious diseases like HIV, TB, schistosomiasis, and so on and so forth. So today I'll be talking about something which I call, what's going on? This is so slow. Yeah. Okay, I'll be talking about schistosomiasis today. So when you look at Africa, this map of Africa, what you see generally is, come on.
That one, for some reason, is lagging. Online, they already see the map of South Africa.
Really?
Yes. You can look here and see what they see.
Okay.
Okay. Yeah.
But you guys don't see it on the screen.
Yeah, no.
Now we see the map of Africa.
There's sometimes delay.
Oh, is it? Okay. So I'll just.
That's one set to one.
Okay. All right. Okay. So what you see basically in Africa, schistosomiasis, basically to start with, is a neglected tropical disease, and it is a disease that you find when there is inequalities in, you know, sanitation, inequalities as in poverty, and all over Africa, we know there are these inequalities, and it is a disease that actually you get from riverine areas, you know, in places where the water of those places has been infected, and this disease is actually caused by three important worms. The first one is called Schistosoma mansoni. The other one is Schistosoma hematobium, and the third one, Schistosoma japonicum. But there are other forms of the same causative agents of these diseases. But what you see here in Africa generally is that you see that the two important forms of these causative agent, which is Schistosoma hematobium and mansoni, you see it around almost the whole part of Africa, where you see the other one plus intercalator, which is a smaller one at DRC. But what is very important on this map is to see that in some part of Africa, which is in Morocco, Algeria, and Tunisia, the transmission of this infection has actually been erupted. So how did they do it over there? So what people from studies, they actually apply what is called the wash technique. That is, that's a technique with which, you know, people get to look at the water bodies, treat the water bodies, and then that helps in breaking the transmission of the infection. But moving back home, if you now move back home, what you see in South Africa, it is actually not a disease that, you know, that government spend more money on, because it is not HIV, it is not TB, and it's not malaria. But what you see is that according to Magaiza 2015, he and colleagues found out that there is 4.2 million infected people of this disease in South Africa, and these people are located within the provinces of South Africa, especially within Limpopo and Mpumalanga province. And what you see in Limpopo and Mpumalanga province, because these two provinces borders neighboring countries of Mozambique and Swaziland. Then you see some form of this infection in KwaZulu-Natal and Eastern Cape, because these are provinces that borders water bodies. And also, it is a traveler's disease. And because of that, you see some forms of it in Gauteng and also in the Western Cape, because you see a lot of people come from the various provinces to Western Cape, Cape Town, and also to Gauteng, which is Johannesburg, you know, for employment opportunities. So, and in 2021, the Boni also found that the prevalence has actually increased from 4.2 to 5.2 million. So it is a big issue in South Africa for us. And not only that, how did this infection comes into being? So how did it worm? How does it grow? How do you get this disease? So what you see is that, number one, first of all, the infection starts from the boy who is hopefully infected, defecating openly into water bodies, and eggs from this worm goes into the water bodies and then grow into the juvenile worm, which is called Miracidia. And Miracidia then penetrates this worm, the freshwater worm, and in the snail, you see the growth of this Miracidia becoming mother sporocyst, and then growing germinal balls, and then grow daughter sporocyst. Once the daughter sporocysts are grown, they are then released back from the snail into the water bodies as Sicaria, which then penetrates the human skin or penetrates the naughty boy who is busy swimming in an open infected water. And also, they then go into, and in the boy who is swimming, it grows into the adult worm, and the adult worm then mates together to now release eggs per day. This worm releases nothing less than 5,000 eggs. And the infection is it is not having schistosomiasis that is the problem, but it is the release of the worm, just a single egg that gets trapped into the human body or into any of the organs of the body is what causes the infection. And we are interested in it because of the fact that these eggs has this ability to cause various forms of cancer. And how did that happen? I'll explain as I go along. So what you see when this happens is that you get the acute schistosomiasis, where the boy who is beaten up, whoever passes through that infected water, you get what is called a swimmer's itch, which is just like you itching your body, and after like a day or two, the itch goes away, and then you get what is called Katayama fever, which basically gives you like a sort of, you know, elevated temperature, and immediately after that, even without any medication, the infected person becomes better. But the fact is that the worm can live in human beings for 30 to 50 years without dying if it is not treated. And there it causes what is called chronic schistosomiasis. And our interest is actually also in what is called genital schistosomiasis, because in genital schistosomiasis, it predisposes the sufferers to HIV/AIDS. And how does that happen? What you see is that from studies, we found that 17% of HIV-infected people also have schistosomiasis. So they actually co-infected. And and schistosomiasis predisposes people to intensive loading of the HIV virus, and also makes the risk of transmission to be very high. How does that happen? It happens in the sense that when the infected person is not treated, what goes on is that the worm then create enzyme which causes inflammation in the genital organs, and from there, especially in women, you have what is called sandy patches being formed. And when these sandy patches are being formed, what you see is that you get aberration of the genitals, and that basically, predisposes people to when people have unprotected sex. Also, I also spoke about cancer. What you see is that the eggs, when they get trapped into the hepatic portal system of the infected individual, what happens is that they generate N-nitrosamines and also 8-deoxynitroguanides, and these are molecular signatures of cancer. Hence, this infection predisposes its sufferers to cancer of various morbidities of cancer, especially cancer of the bladder. So, especially cancer of the bladder. Since 1970s, this infection has been treated with this drug, which is called praziquantel. And praziquantel is actually from the family of that medication we call betricide, and it's been used for the past 5 decades, and it's got 60 to 90% efficacy. But recently it has been deemed unsatisfactory. Why is that? Number one, so the use of mass administration of this medication to a whole lot of people has caused what we now call drug resistance, as the worm has started becoming resistant to this infection. Also, another thing that also happened is that since 50 years ago, there hasn't been that elucidation of the mechanism of action of this medication called praziquantel. And what you have in literature is various people just proposing or postulating that this plays a role, that plays a role, and with no study has really shown the mechanism of action of this drug. Also, it is also deemed unsatisfactory because praziquantel do not have effect on the juvenile worm. That it waits. It's only active against the adult worm, and that is when the adult worm will have already laid the eggs, and the eggs has already gotten trapped in the human hepatic system. Hence, you are treating the adult worm, but the juvenile worm is still there. And also, lastly, this medication do not break the transmission, in the sense that people who have been infected and be treated can still be reinfected again. So can still be reinfected again. And we've got the first report of infection was actually from Fallon, from his study in Egypt and Senegal, where he showed that there have been some pockets of resistance of the worm to praziquantel. And that was in 1998. And in 1999, Ismail also, from his study from Egypt, also showed some resistance. And what we now see that in 2009, the study from Kenya shows that not only do you see this in the rural areas, the resistance in the rural areas of Kenya, but also in the urban areas. So this calls for attention in the sense that we need to find a way to actually develop new the treatment drug for this infection. So when we started looking at this disease, there was this noise about antimicrobial peptides. And why is it? In the sense that people believe antimicrobial peptides are peptides that have short-chain protein molecules, and they've got various, charges which makes them very reactive. And so we also got onto that bandwagon, and the first thing we did was to read around the use of antimicrobial peptide and how they can be used in making new drugs or in designing new drugs. And that we published in 2014 in this paper. 2014 in this paper. And then we now went into using bioinformatics to now identify druggable protein targets in various schistosomal disease, species, and that we published in this paper, Scientific African. And it is the work of these students of mine, Dr. AfLS, who has now found a new home in the US. So he did a whole lot of work on here, and here we discovered, or we actually identified 6 antimicrobial peptides, and we are still trying to see if we can derivatize these antimicrobial peptides and then test them in vitro against this worm. Then we can now say for sure we've got something that we really can move forward and think about patent, just like Simon just said. So now, one of the endearing factors of parasites, generally, when they are in any environment that they do not, that doesn't favor them, is to basically upregulate 2 types of proteins. The first one is the heat shock proteins, of which everybody knows, and heat shock proteins has been studied over time. But the other type of class of protein, which they upregulate in response to whether nutrient starvation, high salinity, drought, extreme temperature, toxic chemicals in the form of drugs and medication, is what we call universal stress proteins, USPs. And we are interested in universal stress protein. And because we know that schistosomiasis is an environmental disease, and also we also see that when you target drugs or anything which is toxic towards any parasite, they upregulate universal stress proteins. So we actually came up and we wrote our first review on this, which we call universal stress proteins as new target for environmental and therapeutic intervention of schistosomiasis. And we went further, not only that, but to look at, we took various sequences of universal stress proteins in in schistosomiasis and other diseases. And we came up with this classification and functions. And I would like you to just look at this class formation. That is, universal proteins can be classified into class 1, which are actually non-ATP binding, and class 2, which is ATP binding, class 3 and 4. And these various classifications is just the domain configuration of the various universal stress protein. And these are the functions which we were able to glean from literature. And they actually play roles in oxidative stress defense, ion scavenging, motility, adhesion, and cell aggregation. So now let's get into it properly. Our collaborator in the US, Rafael Sokwe, who is a girl who does bioinformatics, actually published this paper in 2013. And before he sent this paper out, he called me and said, "No, we just found a universal stress protein, and I see you are interested in looking at universal stress protein. I don't do your kind of biology. So if you can actually see if you can solve the structure of this protein and maybe possibly do what is called structure-based drug discovery." So we took up the challenge, and what Raphael and his student Andres Umba found was that they found that there are 2 universal stress proteins that are upregulated in the 3 major schistosomal worms, and those universal stress proteins play a role in the sense that they help the worm to respond to unfavorable conditions. And one of those universal proteins is called G4LZI3 protein, and which is found in S. mansoni. So we took up the challenge, and the very first thing we did, because we are biologists, the first thing we did was to clone that gene. We cloned the gene into, I'm sure I won't bore you with this because I know most people are not biologists here, but we cloned the gene and we cloned it into a protein expressing vector called PQ30. And this just shows you the digestion. When you clone the gene, then you digest, you cut it, and then you want to know if the gene is running at the right size. And we see that the size of the gene, which is 55 base pairs, is actually here, and this is the vector. So we were successful. We have a successful cloning, and from there we then move on to express the protein in bacteria. So we have a bacteria setup where you can now take the clone gene and express in bacteria to then express the protein of interest. And the protein of interest is at 20.3 kilodalton. This is a small scale and this is a large scale. And because already we do not have anything to characterize this protein to actually confirm if indeed what we are seeing is our protein. So we did what is called a Western blotting. We use Western blot to blot using an anti-His antibody, which basically will find the protein and give us a signal. And the signal is actually what you see here, which runs at 20.3 kilodalton. So, and immediately we did that, we then have to purify the protein of interest, the G4ZI3 protein of interest, away from other bacterial protein. And then we subjected the total bacterial protein to affinity purification. And then this is just the chromatogram, and this is the almost pure protein of interest. But what is concerning is that we see a little bit of residual protein at this level. So based on that, we then move on to do what is called gel filtration. And gel filtration is to actually purify the protein further. And so we purify the protein further using gel filtration. And this is the chromatogram of the gel filtration, or what is called size exclusion. And the protein still runs at the right size, but again, we still have this lower band, which we don't really understand what it is. But because of that, and because we wanted to solve the structure of this protein using X-ray crystallography, so we want to have the protein to be a homogeneous protein. So we then said, we do not know what this is. So we cut out this band and we send it off for mass spectrometry. So when we send it off for mass spectrometry, so what we see is that we saw this 20.3 kilodalton. So the protein is actually the right protein, and then we then characterize the protein using FTIR. And what FTIR gives you, things that is marked are the fingerprint of the functional groups in proteins at their characteristic functional group that you see in protein, and that is what we saw in the protein. So we also characterize the protein further using differential scanning calorimetry. And what this tells you is that at what temperature would the protein denature? So what you see is that the protein is stable up until almost 110 degrees centigrade. And above that, the protein starts undergoing denaturation, and you cannot use it for anything again. And we also did fluorescence spectroscopy, and that is basically to be able to see at what wavelength will the protein maximally absorb light. And we could see that at about 330 nanometer, the protein will maximally absorb light. We put all this characterization and cloning and purification, everything together, and we submitted that for publication, and it was published in this article, which is shown. Okay, then having characterized the protein and know that it is the right protein, the next thing is then to then crystallize this protein. So, and to crystallize, there are two steps. The first step is to form the crystal, and the second one is to test the crystal using this X-ray generator. Okay, so this is the robot which is actually at Trevor Sewell's lab in UCT, and also the X-ray generator. And these are the, I will have to run through this now. It's just a pity my time. And these are the crystals. This is where we started from using the robot. And then we got this, and then we use this as seed stock to now see. And this, from these crystals, these were the crystals that we now sent. We now sent to Diamond Light Source in the UK. And this is just to show you, these are the dewars in which our crystal is placed, and this is the arm over there where you put your protein somewhere, and then you just, and then where you collect your data from. So this is just to show you the beam line that was used at Diamond Light Source. And then this is the data that we collected, just like Emmanuel was saying before, that they had 3.6, we had 3.05, and the resolution was not that good. We tried all we could with this resolution. Just to make it better, we went back to the electron density map. So this is the electron density map we started from using MR Rosetta. We could see that a lot of these are not filled, they are missing. And then when we also did refinement, we tried to refine it further. So we got the refinement done a bit better. And then finally, we came up with this.
We see it.
Okay, we came up with this. And what you see here is that you can see we came up with the partial structure. This is the structure of the protein. And you could see there is this missing part of the protein structure. And this basically corresponds to this part of the electron density map. There's nothing we could do to help it. We tried all we could. We couldn't find this. And remember that we said we wanted to use the structure to do structure-based drug discovery. But because we could not get a total or a complete structure, we have to change that. And then we changed that. We said, okay, let's go back to, we quickly publish this so that before someone else could publish it, we publish it in this paper. And then we changed that to say, let's move on to the next thing to do. But before we move on, what we now did was to now co-crystallize, because we couldn't get the full structure. So we then said, let's actually set up an assay. So in the process of setting up an assay for this, so my collaborator just said, why don't we just co-crystallize this protein in the presence of ATP, and since we said USP can either be ATP binding or not ATP binding. So we co-crystallize it in the presence of ATP, and then we found that, you could see, this was actually very nice crystals, and that is where we are. We are yet to collect data from these crystals. But meanwhile, we changed that, and then we move on to use bioinformatics. And what we did was to adopt the method of Anandi and co-workers. And what Anandi did was to look at polyphenols, but they did their study in TB. So they used natural polyphenols, dock natural polyphenols to a particular protein in TB, and we adopted the same method. And what we did was to dock exactly 10 natural polyphenols to the G4SZI3 protein. And when we docked it, then what we saw is that the docking, or the docking, the docking result, we actually prioritized just 5 of them, which give us very good docking score. So, which is avenatide and so on and so forth. So, and then we take the complex of the protein and the polyphenol complex, now conducted molecular dynamic simulations, and this is what we see. Because of my time, I'll just run through it. We did RMSD to look at the APO. The APO is a protein on its own without any of the 5 polyphenols. And you could see when you do the post analysis, the post MD analysis, the RMSD shows you that quercetin, catechin, and dalbegin, are the ones that actually deviate more. And then we also did what is called the RMSF. The RMSF looks at the fluctuations of the various parts of the proteins, and that is what you get, various parts of the protein, so that when the docking happens, which are actually fluctuating, not stable. So what we found also is that, again, it's changed, okay. So at the end of that structural flexibility, we found that catechin, darbegene, and quercetin gave us a better result. And then finally, we looked at what is called ROG, which is radius of gyration, which tells you about how when the polyphenols bind to the C alpha of the protein, how did the C alpha behave, the behavior or the deviation. And what you see here, this is the apple, and then you can see curcumin, catechin, and so on. But what we finally did was to now do what is called MMGBSA. So this is a technique which talks about the binding free energy, and those with the better binding free energy are catechin and curcumin. So these are the polyphenols. So at the end of the day, we came up with this model where, you know, we did 100 nanosecond MD simulation, and for these two systems, you can see how over time, how the polyphenols integrate itself into the active site of the protein, you know, as you go along the simulations. And with that, we then came up with a model which we say that we came up with this model, which we say that the presence of polyphenols, basically, this is the one which gives all these free radicals, and when they're releasing reactive oxygen species which form these adults, but in the presence of polyphenols, because we know polyphenols as natural scavengers of free radicals and reactive oxygen species, so it blocks those pathways. For example, it blocks this pathway here, whereby you see, you do not have DNA strand breaks, and when we know when there are DNA strand breaks, what happens is cancer, because it releases the 8-oxyhydroguanine, which I talked about, which is a cancer signature. So along the line, what we have found out from our study is that, looking at this study, we can actually add polyphenols as an additive to the praziquantel. And because basically this drug is used to treat school-going kids, so this can be added and be made into an ELISA for these school-going kids. So we also published this work quickly in this paper, which is actually shown there. And then we got a request from a collaborator in Belgium to look for those molecules that can actually activate and not inhibit. Because every time when we talk about drug molecules in the biosciences, we are always looking for something which inhibits the protein. But we don't always look for things that actually activate a particular part of the pathway. So we then got that request from our collaborator in Belgium, and then we, together with this student of mine, Dr. Mabunga, so we wrote this review article which talks about protein-protein interaction modulators, not inhibitors now. And then we started searching using our protein of interest to search for other partners that may actually play a role. And with my postdoc, Dr. Masamba, she took our protein of interest, which is the G4SZI3, and used this program called STRING, and we found two proteins. We actually prioritized these two proteins. This one we prioritize because it is a heat shock protein 60. I remember from my introduction, this is one of the class of proteins that parasites upregulate. And the second one, which is the SMD, is a class radiatory phosphate dehydrogenase. And what is the role of this what we call GAPDH? Now, what GAPDH does is that it plays a role in glycolysis. And from our basic biochemistry, we know that glycolysis at this point it is this point at which energy is generated. And what you see is that in glycolysis, that enzyme catalyzes this reaction from glyceride 3-phosphate to 1,3-bis phosphate glycerate, releasing NADH, which goes into the electron transport chain to generate energy. And also, at this branch point also, we also found another protein which is called TPI, triphosphate isomerase, which also can then be converted into glyceride 3-phosphate dehydrogenase to also give energy. So hence, we think that the worm gets its energy from using the glycolytic pathway to generate energy for itself. So, and we came upon this paper, which all have solved the structure of GAPDH, and they said the energy generation by GAPDH is used by the worm for mobility and also migration of schistosomolar and also to produce the eggs that also caused the problem. So what we did next was, I'm about to round up. I know my time is spent. So what we did next quickly with this PhD student of mine and the master student, we then clone and express these proteins. And after we've cloned and expressed this protein, we conducted what we call, we did interaction studies just to see if indeed there is interaction between the two proteins. And if you look at the protein complex here, which is the green, this, you can see that the UVV spectrum from the protein, from the NAD binding domain of GAPDH, it's much lower when you basically interact the SMTPI with it, that is the triphosphate isomerase. And the reason is just because that there are no binding space left for those, what do you call it, those aromatic side chains of the protein to be able to absorb more light, and hence you get a lower absorption. And we also did Raman spectroscopy just to see if at all this interaction that we see. So Raman spectroscopy was done to actually look at the qualitative way. Is there any, if you look at here and then you come here, you could see a little bit, but here you can see that there is something which is moving here. These are just new results which we just got last week. So we are still processing the data as we go along. Then finally, we now use microscale thermophoresis. And because of my time, I'm not going to explain this, but I'll just tell you the result which we got from this. So this is the result. And what do we want to know? We want to know these two proteins, do they interact? If they interact, what is the strength of interaction? So this is the TPI, and this is the TPI and the NAD binding domain of GAPDH. And when we did the interaction, we found that the interaction, which is the KD, is actually 2.3 micromolar. And the stoichiometry is at 2. So basically, according to Weinberg and Quinn 2006, so they said any interaction in the region of 10 to the power of -6 is actually moderate. So what they said basically is that what we can take from this is that these two proteins interact, but they have a moderate interaction. The interaction is actually not strong, it is not covalent. So based on that, we then, because we know that there is interaction, we have now established there is interaction. So we did our bioinformatics studies, and then eventually we came up with these 3 druggable compounds. So which we named UJKMA1 to UJKMA3. And this we just sent out for publication this Monday. So these are the 3, and we hope to actually derivatize more compounds from these 3 parent compounds so that we can then test it against the worm. So the last part is that, which we talked about, is that there is no mechanism of action elucidating the effect of praziquantel on the worm. So what most people have actually postulated is that divalent cations play a role in the way that praziquantel worm. And Martin in 1997 postulated that, that what happens is that when praziquantel comes into contact with the worm, what it does is that it actually disturbs it actually disturbed the calcium homeostasis in the worm. And in 2005, and in 2005, Mendoza Silva, working from Brazil, and his colleagues also showed that calcium plays a role. And also the study of Umba et al. Also shows that divalent cations plays a role. But remember, these are all bioinformatics studies. They have not been proven. So we now said, let us prove this. And together with these master students of mine, Elite League, we then selected 3 divalent cations. Calcium, which they proposed, we selected magnesium and also zinc. And what we did is that we now did MST on all of this, on all the 3 in the presence of the protein. And what we found is that all the 3 selected divalent cations actually plays a role. They actually interact with the protein. With calcium, it is 0.1 micromolar. So why do we have micromolar? It means that the interaction is moderate. And remember that this is an enzyme. Hence, those divalent cations act as cofactors to this enzyme just to help the enzymes or the protein to carry out its function. And I think this is just the result we got just this past Friday. And we've not, and there are so many other results, but I think I'll stop here because of my time. And lastly, I would just want to acknowledge, those I work with, whom I've been working with for a while now. First person is Trevor Sewell, which we've been working together for a long time, and our collaborator at Oxford, Frank Von Deft, who basically, in fact, also is so happy to have one of my students still in Oxford now, doing some structural work there, and also funding from GCRF. They gave money to Africa under this program called Synchrotron Techniques for African Research and Technology, called START. And through START, we are able to use the diamond light source for all our structure, and then funding from South African Medical Research Council, NRF, and then acknowledge my former university where I started this work, and University of Johannesburg, where I am now. Thank you for your attention.
Thank you very much, Professor Kapo, for the interesting talk. So we've already gone into tea time, so I think we have got time for one question. Okay.
Maybe make it 2. Mine is simple. Maybe make it 2. Thank you, Prof. Kapo, for a very impactful presentation. I just got interested on the robot. I want to know, I missed it. I want to know, how did the robot contribute into the work?
Oh, okay. You're asking about
the robot.
What we did is that the robot is used to set up your crystallization, because with crystallization, you've got to pipette, so you have to do a lot of pipette, and sometimes you may make mistakes. So the mosquito robot, which is used for crystallization, you put your protein in there, you know the concentration, you set up the values on the robot, and in there, it pipettes the robot. The robot pipettes the volume that you need, and then put it in your 96 well plate, and then you can now put, set up your crystallization. So you don't have to do anything. You just need to calculate, the concentration of the protein you want, the buffers that you want to set up. Everything is programmed on the robot. You just put your protein, your pure protein there, and then it's pipettes for you, and then you can just put the setup, the plate into the 4 degrees, and then you wait, and every now and then you go check under the microscope to see if you have crystals forming or not. Yeah.
Nice. Did it do a great job?
It did. It did a very nice job.
Okay, we have one question from online. Bongani will read it out to you.
Okay.
Yeah, I think it's Dr. G. Yeah, it's a yeah, he had to he had to leave drop off. The question is, do you have any insights into which amino acids permease might be responsible for stress survival? I see the potential avenues for collaboration here. There was a second one, perhaps quickly, maybe you may not respond to it. Did you try using an AlphaFold model of G4LZ13 to address the missing density? If not, was it due to AlphaFold not being available at the time? Please feel free to share our email. I'll be sure to reach out. Thank you.
Yeah, okay. I think because of our time, I won't answer that question, but I will reach out to Emmanuel, yeah, and then we'll take it from there. Yeah.
Okay, thank you very much, Prof. Can you please give him another round of applause? We have tea time now. The time is
it's 20 minutes.
Sorry,
in 20 minutes.
Lawrence is listening now.
Oh, can you just ask,
Lawrence.
just in case, are the other two speakers in the room?
so I know there's a Lorato in the room.
So we need what's it, Malebo and Ekain Etape.
Are they they're not online now. Yeah. Oh.
Okay, then
You can take Lawrence's question then.
yes, so we can then take Lawrence's question and then we can go for tea.
Okay. Lawrence, can you please ask your question?
Yeah, I just wanted to give Professor Capo an opportunity to talk about the how the Federation of African Society of Biochemistry and Molecular Biology was able to land the global IUBMB conference in 2027 in Cape Town, and that goes into how we can all synchronize our calendars and have a molecular bioscience, structural biology footprint at that meeting.
Okay. Thanks, Lawrence. Yeah. I think I'm supposed to give that talk at the Science Forum on the 3rd or 5th of December. But I'll just give a quick overview. What we did, we were able to land that it's actually not FASBMB, the Federation of African Society, that landed that conference. It was actually the South African Society for Biochemistry and Molecular Biology, when I was the president of that society. So what we did was that so that it's not just very muted. They couldn't hear you. Sorry, Lawrence, can you hear me now?
Yes, I can hear you now.
All right,
I
sorry.
put into the chat the link to the Congress So I don't know if you want to repeat everything you just said, but I put into the chat a link to from the IUBMB page, giving
Yeah.
the 2027 dates.
Yeah. So yes, we basically will be having that Congress in 2027, and we've started mobilizing from Africa. Like I just mentioned during my talk, that when Emmanuel was talking, we just had our first BMB, the Federation for Africa Societies Congress in Nairobi, Kenya, this past 2.5 weeks, and it was very interesting to find out we have people coming from Tanzania, we have people from Ethiopia, we have people coming from, even somebody came from DRC. So we have about 14 countries represented at the Congress of FAST BMB. So which gives us, that hope that we surely will get, that representation. Obviously, South Africa, Zimbabwe, Namibia, we are all represented, and also Zambia as well. So our job now is to make sure we advertise and market this, throughout Africa. And we will be having an LOC meeting, I think next week, where we're going to talk about strategy to market this. But very important to me is that we would like to have subsections of, like we've talked about biochemical education. We're looking at people in science education, somebody who can lead the biochemical education side of things. We will look for somebody to lead the biophysics side of things. We're actually thinking of what's his name? Again, Kruger, Taj Kruger. And we're also looking at people who lead various sections over there in this. The good part of it is that the young scientists in Africa are not going to be left out. So the parent body, IUBMB, is happy to pay for 120 young scientists to attend this Congress. 120 to attend. And also, FAST BMB is also going to sponsor young Africans to the conference, where we pay their flight, we pay their accommodation throughout the duration of the Congress. So I'm just going to say, go to the website, IUBMB website, and you will surely see, the exciting things that are there, and also the various funding opportunities that are available over there also for young scientists and early career researchers in Africa. Thank you. Did I answer your question?
Very well.
Yes, sir.
Yeah.
Yes, thank you.
We also question from Prof. Shane.
Thank you for the very nice talk. I'm actually very impressed with your talk. And I also feel a bit depressed based on your very first image in terms of how the disease would circulate and so on. And I can see that even though we all love to have African libraries and to help solve some of the health issues, but there must be some fundamental clean water and these things that has to be done. So I'm wondering whether one could, to go further with African Light Source initiative, one could reach out to public service type of government, public health, and try to get those people together to have a common goal to solve both the science side and the infrastructure side. I believe that will help strengthen the African Light Source initiative. That just came to my mind. Yeah. Thank you for your talk. Really appreciate it.
Thank you so much. I agree with you. Yeah, thank you so much. What I'll say is I think we need science advocacy in Africa. And from the side of the African Light Source, like Simon and others are doing, I think possibly when we speak to these African leaders, we basically also need to put something up to tell them, in fact, there is need to actually upgrade the infrastructure that we have, especially that of sanitation. that we have in Africa. I think it's really, like you said, it's very depressing, what you see. And in most countries in Africa, you find that this infrastructure are not there. So, and based on that, I mean, at this age, you still have people doing open defecation, into water bodies, which is really depressing. And these are the type of things that, I mean, those who interface these leaders should basically, push forward. Yeah, I agree with you. Thank you.
Okay, can we please give Professor Kapo another round of applause? So we've eaten up quite a bit of the tea time. So I'd implore everyone just to pour a bit of coffee, enough for a sip, and then we come back very quickly. We come back at 11 o'clock.
middle.
Have to be on this window to make it work.
Yeah, so if you need to go and switch something, then just do it quickly.
Okay.
All right. Morning, everybody. I am your chair for this session. I'm Wilson from UCT. The session, we've got 4 exciting speakers, range of topics from imaging to structural biology. I'll bring the first speaker, Skuda, from ID or I-13 beamline at Diamond Light Source. Thank you.
Good morning.
Okay, 25, 25. Right. Then. Okay, am I sharing?
Thank you, Wilson, and good morning, everyone. So, as Wilson has mentioned, my full name is Kuda Kwashe Jakarta, but I think everyone just calls me Kuda, and that is fine. So the title of my presentation today is Tachography and Bragg Coherent Diffraction Imaging at the Diamond I-13 beamline, and I'm going to give a bit of highlights. So I work at the Diamond Light Source in the United Kingdom, close to Oxford, about 16 miles away. So this is an aerial view of the Diamond Light Source. As you can see, when you're looking at the roof, you have all these solar panels, which is something that is relatively new. I think they were already putting in the solar panels when I joined the Diamond Light Source in 2022, like towards the end of the year. Which actually helps a lot because it's provided a big saving in energy, because one of the biggest sort of costs of running a synchrotron is the electricity. So I think we could probably be thinking the same way when it comes to the African light source. So the Diamond Light Source is a 3 GeV synchrotron, and of course we get a very high flux of X-rays and high brilliance. And it's an expanding facility. And additionally, I think we have close to about 1,000 employees working at the Diamond Light Source, beamline scientists, engineers, technicians, and everyone is required. So I think we have already heard a lot about 4th generation sources and the upgrades. There's the EBS, ESRF, Max 4, and Sirius. They've already upgraded. So Diamond has already it's been proposed and approved. They're going to upgrade to. There's going to be an upgrade which will provide the opportunity to exploit much brighter X-ray photon beams and an increased coherence over a large energy range. And what's also going to happen is with the proposed Diamond 2 upgrade, it's going to provide the possibility to add 5 new beamlines. So at the moment, 2 are planned to come with the upgrade. And then afterwards, over time, there will be some additional ones. And of the 2 that are proposed, one is spectroscopy beamline, and the other one is an imaging beamline. Okay, so at the Diamond Light Source, there are about 33 beamlines at the moment. And of the beamlines, a third of these are for imaging and microscopy. And this gives you an idea of some of the beamlines like I-12, I-13, I-14, I-08. So all dealing with various types of imaging and microscopy. I-12 is your full field tomography beamline. So if you have a large sample, like a fossil, and you wanted to do tomography, you would probably want to go to I-12 because it also goes to high energies, I think around 120 kilo electron volts. There is actually an iconic South African fossil specimen that you guys probably saw yesterday. In the vault, there was the little foot specimen. So the skull of Littlefoot, I was still working in the Evolutionary Studies Institute when it was fully excavated. We tried to scan it, and what was evident was that the inner ear was well preserved, but of course in a lab-based source, because it's too dense, you couldn't get images with the right quality to actually study that. So it was then taken to the I-12 beamline, where they did tomography, and because the diamond light source is also next to the neutron facility ISIS, then they also did some neutron imaging, and the papers have been published. Additionally, there are also some electron microscopes at the diamond light source, which are used for imaging. Okay, so I work at the I-13 beamline, and at our beamline, so it's got two beamlines, but with, of course, we've got two different insertion devices. And so you get the two beams. One of them is called I-13-1, coherence beamline. And then the other one is I-13-2, the imaging beamline, also known as the Diamond Manchester beamline, because it was built in collaboration with the University of Manchester. So on the coherence beamline, we do the sort of tychography and break CDI, and on the imaging beamline, we do mostly tomography. So this allows for multi-scale and in operando imaging. So just a few highlights from the imaging beamline. So here we're looking at just the imaging beamline. So that's this one here. So our beam is over there. These two beamlines are the longest beamlines at the diamond light source. They're about 200 meters away from the insertion devices. And so here, this is where the ring ends. And then, of course, there's a flight tube to direct the beam all the way to the optics hatch, where you have a double crystal monochromator. You've got some slits, and you then have your sample stage and a detector stage also. And there is enough room also to sort of be able to do in situ measurements in the hatch, in this experimental hatch. So on the imaging branch, the energy range, you can go from 8 to 30 kilo electron volts. The sample sizes that you can scan between 10 millimeters to 50 microns. And the resolution you can obtain is between a micron to 50 nanometers. As I've said, you can do full field tomography, and that takes just a few minutes. It takes, depending on the sample and what you're doing, it can take probably a few minutes to a few seconds to acquire a data set. So this is what it looks like when you're actually doing the experiment. So here we have our detector with the different objectives. So you can, depending on what resolution you want, you can change the objective that you're using. And over here, we have our sample. And in this particular case, this was an in situ experiment where the sample was being heated at the same time. So here is the heater. I think in this particular case, Roberto, who was the scientist, the PI for this experiment, he had a heater, and you would blow some air towards the sample so that you achieve the temperature that was required on the sample. So and the sample, of course, is sitting on a rotation stage with some translation motors below. Okay, so additionally, on the imaging branch, what you want to do is you want to do sort of, because as I've already mentioned, it takes like a few seconds, sometimes just a few minutes to acquire one data set. But if you then want to do 300 samples, you'd have to open the hatch, get in, change the sample, and put in the next one, and it becomes too, what can I say, tiring, and especially late at night, the chances of making mistakes become higher. So there is a sample changing robot which allows you to automate the whole thing. So you put your samples on a specified location, and the robot just picks this next sample, puts it on the rotation stage. You do the tomography, it removes, puts it away, takes the next one, and puts the next one, and so forth. So this allows for fully automated scanning. You can do more than 300 samples per day. or more. And all you need to do is just sit in the control hatch and monitor the experiment as it goes on. And that also allows you to get more statistically relevant data, because if you can do more samples, then you can get more statistics and sort of get more reasonable results, I think. So, which is also, I think this is also a new standard for static samples. A good example of a very recent experiment, which was done this year, I think this was around just before August. Some of the researchers from Belgium, they brought this wood. They were doing quite a lot of wood specimens, and what they were trying to do is to identify different wood species from Africa. And as you can see here, the quality of the images was just seriously amazing. And I think some of these images are now being used as desktop pictures in our beamline. Okay. And then another example of an experiment which made use of also the sample changing robot is the imaging of these human placenta samples. So the idea behind these experiments was that every 16 seconds, there is a stillborn child. And every year, there are about 2 million stillborn children. And what these guys are trying to do then is they're trying to image this placenta, and using 3 different modalities. They're using MRI, they're using computer tomography, and one other technique which I can't remember at the moment. But the idea is then to put the information together and then try to sort of fully understand what is happening in the placenta, so that they can then sort of at least try to prevent stillborn births. So this, as you can see, this is an image of one of the high quality image here in one of the samples, and you can actually see some of the blood and some of the blood vessels and what they call villi networks inside the placenta. Okay, and then I'm not going to say a lot about these Talbot interferometers, but because there's someone else who's going to come and talk about these Talbot gratings. But also, this is sort of another sort of side to the beamline. There is a lot of use of the Talbot interferometers, where you get your beam on the falling on the sample, and you put a phase grating, and that causes some interference, and you get an amplitude grating. And from this, you can then get the transmission, which would be the dark field image, the phase contrast image, and the absorption image. And once you have these 3 imaging modalities, it allows you to to actually see more clearly, especially with the phase contrast, what is in your sample, especially when you have soft tissues, soft tissue materials. The idea is at the synchrotron facilities, when you're doing tomography, you have the possibility of using inline phase contrast. So when you're using inline phase contrast, you put your sample over here on the rotating stage, and depending on the energy of the X-rays that you are using, that determines the distance at which you can put your detector. And then you can then sort of get phase contrast. But the phase contrast that you get is not, you can't quantify it from the reconstruction algorithms that are available at the moment. But then if you were to put in some gratings, you can actually quantify the phase that you get from such. This thing is no longer working. Okay. Yeah. So here is, for example, what you'd get. This is a bee. A bee's. You're looking. I think these guys wanted to look at the retina of the bee, so you can see the eyes here and just below the eye. And here, this is the absorption image. As you can see, the contrast is not that great, and you're looking at the differential phase contrast image and the dark field image. And once you then reconstruct in 3D, you get these 3 slices. And if you then put a line profile along these 2, the absorption image and the phase contrast image, you can see actually the intensity profiles. And from these intensity profiles, it is very clear that from the phase contrast image, it is easier to actually distinguish the different sort of parts of this sample. So this is what's happening on the imaging branch of the beamline. Now, when we go to the coherent side, which is the I-131 beamline, which is where I work at, this is actually a picture of what it looks like inside the hatch. So here we have a robot, and on the robot we have a photon counting detector. At the moment, this detector here, this is a detector which is called Excalibur. So this Excalibur detector is the one that is sitting inside the robot. So and over here we have a different detector, an IGA 500K. So the sample is sitting over here near this sort of blue thing here. This is where we have the sample. So what we do here is we do two main techniques. We do tachography. When we're doing tachography in transmission mode, then we get our beam onto the sample here, and after the beam passes through the sample, we've got the flight tube, which is under helium, and the X-rays pass through the helium flight tube, and they're detected over here. So the sample to detector distance is up to 15 meters. Of course, you can vary. You can also get less if depending on the experiment that you want. And then on the robot, the detector here, we use it for break CDI measurements because you then need to move the detector around in the 3D space to look for these break diffraction peaks. So the characteristics of the beamline, energy between 7 to 24 kilo electron volts. It's a fully horizontal layout. We've got slits at the source, deflecting mirrors, silicon, platinum, and rhodium. We've got a monochromator with silicon 111 crystals. Sample to source to sample distance is 220 meters, and we've got spatial coherence of about 400 millimeters, but it's variable with the front end slits. So if you can reduce the slits opening, or you can increase it, and that changes your coherence length. Okay, so I have been talking about tachography, but maybe to explain what tachography is really about. So in conventional microscopy, what you do is you get your radiation incident on your sample, and then it is scattered, and you then use your lens, and that lens directs your scattered light onto a detector, and then you capture your image. But this image, the resolution and the quality is going to be sort of limited by the type of lens that you have, and the quality of the lens, and the profile also at the higher scatter angles. So the question then is, can we achieve higher resolution? Then the idea is, what if we decided to, instead of using the lenses, we do away with the lens? And so what you then do, I'm going to sort of talk about maybe coherent diffraction imaging first. So what happens with coherent diffraction imaging is you have your beam of X-rays falling onto a sample, and then, of course, what you want when you're doing coherent diffraction imaging is that the size of your beam should be bigger than the sample. And so when the size of the beam is bigger than the sample, after the sample, your X-rays are going to be modified because some of the X-rays have passed through the sample. And in the far field, you put your detector. And when you put your detector in the far field, you get an image that looks pretty much like that, right? And so what you then do is, if you take a Fourier transform of the beam that you have, right, of the probe, you can then you get an image of what this is supposed to look like, right? But then initially you don't really know what the beam looks like. So you start with an estimate of what the beam looks like and what the sample looks like, and then you take a Fourier transform and you get an image. And once you get the image, you compare it with what you actually acquired, because on your detector, what you acquire is the phase. No, you don't acquire the phase, but you acquire the intensity image, which is the absorption image. But when you've done the Fourier transform, you get actually the phase and the absorption image. You can actually separate the two. So once you get that, you compare then between your estimate and what you actually obtained, and then you sort of and then of course what you then know, you do an inverse Fourier transform. If you do an inverse Fourier transform, it is then supposed to you can then update what your probe is supposed to look like and what the sample is supposed to look like. But when you do the inverse Fourier transform and when you're updating, you then put a mask because you know that your sample was smaller than the size of the beam. So so that allows you to put a mask. So that's how you then do this iterative sort of technique, and eventually you then figure out what your sample looks like. So that's what coherent diffraction imaging is like. So effectively then, what you're now doing is you're now doing imaging and without using a lens, but your lens is now sort of the computation here, and that allows you to get better resolution in your images. So with tychography then, it's an extension of this, because what you now have is you've got your beam falling onto the sample, but instead now, because you want to scan a bigger sample, so you no longer have your beam fully illuminating the sample. So the sample is no longer smaller than the beam, but instead the sample is bigger. So what you now do is you scan the beam across the sample, but now the limiting factor, your constraint, is that you have these sort of what you call them. You make sure that you scan over here, and then there is some overlap. Yes, so you have some overlap, and that provides your constraint. And from that, you can then sort of reconstruct what the image looks like. So that becomes then tychography. But then with tychography, at the moment, it's still sort of a relatively new technique. And because of that, there are still sort of that people who are working really hard on the techniques and trying to do bigger samples. And so at the moment at our beamline, you can do sort of around 100 micron size sample. 200 microns is pushing it, but definitely beyond 200 microns, it becomes quite complicated. So the thing is not working again. Okay, so this is what's happening here. So you can see your beam is scanning around. We are scanning here a Siemens star, and this is what you record on your frame. So this is what you'd be doing. So if you have a very thin sample, for example, like I don't know, a few nanometers thick sample, you can then just take these 2D tychographic images. So you acquire these frames, and once you've acquired these frames and you reconstruct, you can then reconstruct what the sample looks like. And so after enough iterations, you get a very, very good picture of what your sample looks like. So that is a 2D image of a Siemens star, which is just a standard sample that we usually use to just check our alignment. But then you can then do tychotomography, because here, this is just a 2D sample, and this only you can only work with very thin samples. What you want to then do is, if you want to see what your image, what your sample looks like in 3D, you then rotate the sample and acquire the same number of frames. You acquire, again, more projections. Okay, you acquire more projections and you rotate the sample, and you acquire more projections and you rotate the sample. And then after that, you can then do some reconstruction first you do reconstruction of each projection, and then you do the reconstruction of the 3D, just like you do in tomography, and you can get an image of what your sample looks like. So here is an example from our diamond beamlines, actually, and partly some of done at Oxford. Cyril Besnar, who was working on this, is from Oxford. He's a postdoc, and he was looking at dental caries in human teeth. So he used TEM, he used tomography, he's also used tychography to image the teeth. And at some point, when you get to higher resolution, you can actually start seeing the crystallized. So something that I forgot to mention with tychography, what is the advantage of using tychography? I talked about the resolution. It allows you to get very high resolution. And at the moment, the best resolution that has been acquired is at PSI. and they've managed to get 4 nanometers. At our beamline, the best we've managed to get is 18 nanometers. And of course, we're working on it. I have very few minutes left, but there's also Bragg CDI, Bragg Coherent Diffraction Imaging. So what you want to do is you want a single crystal material, and you have your coherent beam of X-rays incident on your sample, and you put your detector, this is where you are now using the detector that I was talking about on the robot, and so you get a diffraction. So you put it in the Bragg condition, and in the Bragg condition, you're going to get a diffraction spot at a certain point. And so you put your detector at that point, and then you observe this diffraction pattern with these fringes. And if you then reconstruct these fringes, you get an image that looks like this. And from this, you can then be able to reconstruct the particle shape, and you can also be able to determine the internal strains of your samples. Actually, at our beamline, the software that we are using and implementing for Bragg CDI reconstruction has been written by Marcus Newton. From that discussion we're having about data analysis, we also have some super users who actually then help to improve things at our beamlines. Here's an example of some of the work. Yeah, I'm almost done. I'm almost done. So here's an example of some Bragg CDI, which was done on perovskite material, which was because for perovskites, you want to check, they're used in photovoltaic cells. So you have your incident beam of X-rays, you get your diffraction pattern, but it was an in situ experiment. So you are then illuminating with a laser, and then they were looking at the strain fields in the sample. And we also do XRF. This is some XRF that was done on samples from Fukushima using XRF to see if there was still some uranium in the samples. And we can do XRD. We do methods developments. Where are we going? We're trying to do larger volumes, and to do larger volumes, what we also need is higher flux. So at the moment we use pink beam, and we are trying to go faster. And to go faster, we have I talked about a Dectris detector that we use for tachography. And with this Dectris detector, we're currently using an IGA, but Dectris have partnered with us, and they brought a new detector that they have just launched in August called a Celun. But before they launched it, they brought it to our beamline. It's faster, it acquires more frames per second, and we managed to get to. We are currently the beamline with that can do tachography in the fastest time. Of course, you have to then sort of do a lot of other with the triggering at the beamline. If we can run faster, it will allow us to then be able to do in operando imaging. So because at the moment, when you're doing tachography, it takes something like, it can easily take 12 hours to 6 hours to just do one sample if you're doing tachotomography. But if you could do it faster and in such a small time, then you can probably hit your sample and track the changes as you're doing the measurements. So these are the beamline scientists on the imaging branch and on the I-13 one. Thank you. Sorry, I went over time.
Thanks, Kuda, for the interesting talk. Yes, question?
It's a very simple question. When you're talking about coherent and what? The coherent technique and
Diffraction imaging.
Yeah, and the tachography. Are they the same things with the direct kinematics and inverse kinematics? It reminded me of that. I'm not sure. I'm just asking. That's why.
I don't know direct kinematics.
Or inverse kinematics, the tachography, because you explained that the sample on the coherent was bigger than the beam.
So if you want to do CDI, you want the beam to be bigger than the sample.
Yeah, but
CDI, but then when you're doing tachography, it's because when you're now trying to recover what the sample is like, right? You then need some constraints, and that allows you to put a constraint so that it can converge. But whereas when you're doing tachography, you then use the overlap. to then give you the constraints. And so you can do a bigger sample.
But is it the same as the kinematics, inverse kinematics, the tachograph? It is, ne? Oh, it doesn't know. Oh, okay. Okay, it reminded me of. Okay.
Unless you explain.
Oh, okay.
Yeah.
Okay.
Any other questions? Yes, Prof.
Yeah, in there.
Right, thank you. The most part were more on the beam diamond, so I don't know much, but initially it was quite fascinating for we those who do single crystal diffraction and all that. As I look at some part, it was like my lecture for telling my students how to see an atom, you know, and I tell them, can you see an atom like normally you use your microscope and all that? For amoeba, you cannot do that, but diffraction processes. So quite interesting. The idea about trying to bath your sample completely, you know, by having a bigger beam size compared to the ratio of the sample size, you know. I think we get around it in a way by having the sample itself rotating through that in the old times, maybe 40 theta angle or many angles, so that it's a way by which you bath the whole sample, make sure that the volume of it itself is totally captured, you know, so that you capture all the indices. So I don't know why it is still inferior to the typography in terms of having, in this case, your sample being bigger than the beam, but then you go through sampling through the various cycles. The difference I'm not getting properly, because I think we overcome that by rotating through so many angular variations to be able to statistical lesson of what in the indexes within the compound that must be captured. I don't know whether you get what I'm trying to say. Okay.
But maybe I can explain why. So the reason why we want the sample to be, so it's you want the sample to be smaller than the beam, not the sample to be bigger than the beam, right? And it's more for the when you're now doing the reconstruction to then figure out what the sample looks like, because this is an imaging technique. And I think you are referring to a diffraction technique, right? So I think that is where the difference is. We are doing imaging, and what we essentially want is it allows us to have a constraint, which then for the reconstruction, it allows us to converge.
So that we get a better resolution.
Yes, the technique allows us to get a better resolution, but then for the recovery now, isn't it? If I do imaging, right? So for example, if I do tomography, I have my full beam on the image, and I get and I just get one radiograph, right? But I want a 3D image, so I then have to apply some mathematics and computation to get the 3D image. So for break CDI, what I now want also is I need to get an image of what my sample looks like. But for that now, it's a different technique, because with tomography, it just relies on the absorption of the different materials. Whereas this one, yeah, it's different. Yeah, and so when we reconstruct, that's why we need some constraints, because it's an iterative reconstruction. And for the iteration, you need some constraints. Otherwise, it just, yeah, it never converges.
So it seems like our next speaker is not yet online, so I think we may have more time for questions. Is there?
So she is online.
She's online. Oh, okay. So one final question from the floor, or maybe I can have one from, okay.
Yeah, just to keep it short. Very, very impressive work you guys do there at the beam line. I just want to know if you're able to handle air-sensitive samples. Air-sensitive samples, like if you want to look at battery material, for example, or catalyst, you cannot expose them to air. Are you able to deal with this kind of materials in your beam line?
Yes, we actually, yeah. So a lot of the experiments that we're doing at the moment are batteries in different environments, different situations. So yes, we can.
Yeah, maybe one final question from. Okay, so one final question from online. We've got Philip.
All right. Can
Yes,
you hear
yes,
me?
Philip.
Yeah.
Yes,
All
we can.
Good presentation. And although I'm a bit confused, I need a clarification on terms of the coherence, where you said the size of the beam must be greater than the size of the sample. Because I know in some of our waste stress analysis, we use a low structural resolution. So I want to know the size of your beam for that. That's one. And then the second one is a general question, because you made us to realize that you want to upgrade the system of the diamond isot. And that means that there will be some magnets that you guys are not going to use again. Can it be given to us in Africa, whether we can do something from there? Or what is your, or what can you say? Because you work there anyway. All right.
Yeah. Okay. Thank you very much, Philip, for your question. I think about the magnets. Yeah, I don't know. I don't know what the plan is for the use of those magnets. So maybe I could find out. And then the first question. What size? Huh? Beam size. Yes. What is the beam size? So the beam size that you use when you want to do. So you can decide what the beam size you want, because we have a zone plate, which is like a lens, which allows us to then focus the beam, and then you choose where you want to put your sample, depending on the size of the beam. So you can if your sample is very small, you can even have a 5 micron size beam, or you can have a 10 micron size beam. We also have a KB mirror, and so with the KB or a zone plate, you can then decide what size of beam you want on your sample, depending on the sample size. Thank you.
Thank you. I think in the interest of time, I would like to respectively end the questions here and thank Dr. Kuda. Our next presentation will be offered online. It will be by Carmen Tolmi from the University of Free State. Oh, okay. She has to share. Okay. Carmen, are you online? And if you are online, can you please share your slides with us?
I think she's muted.
Can you also unmute yourself, please?
Hi, sorry. Can you see my screen?
Yeah. Yes, we can see your screen.
I just need to see what I'm doing now. So I just started doing something very noisy here. So can you just give me a moment just to move?
Come in. Are you moving your mouse? I think I don't see your mouse moving.
Hi,
Oh, okay.
can you see my screen?
Yes, we can see your screen.
I didn't just see. Something is not right here. Okay. All right, can you see my slides?
Yes, we can. So ready when you are coming. Thank you.
Thank you so much. Okay, so good morning, everybody. So my name is Carmen Tolmi, and I'm from the University of the Free State. And today I would like to talk to you about the hydrologist production, purification, and crystallization of sterile 24C methyl transferases from opportunistic pathogenic fungi. So this is in collaboration with a number of people, including people from the ARC and people from the University of Oxford. I went here. All right. So as you all probably know, fungi are ubiquitous in our environment, but opportunistic pathogenic fungi are fungi that usually do not infect immunocompetent persons, but they can cause localized or invasive disease in immunocompromised patients. And immunocompromised patients are actually quite a broad group of people. So especially relevant to Africa are individuals with HIV and AIDS. And we have a lot of people in South Africa that are HIV positive or suffer from AIDS. But then there are also a number of other people, such as sufferers of chronic conditions, including cystic fibrosis. If you are a patient in the intensive care unit in hospital, if you have a dwelling instrument, or if you have received immunosuppressive therapies, for example, if you received an organ. Then also during the pandemic, we saw a large number of fungal co-infections, and that increased the mortality rate greatly, up to 83% despite treatment with antifungals. So in addition, the number of invasive fungal infections that are detected is increasing globally. So in 2022, the World Health Organization released their first list of priority fungal pathogens, and they are grouped into critical, high, and medium priority. And in this study, we will be looking at enzymes from the pathogens in the critical group. So Cryptococcus neoformans is found in bird droppings, and this causes cryptococcal meningitis, especially in HIV/AIDS patients. Candida auris is a hospital-acquired infection and is very resistant to antifungal treatment. Aspergillus fumigatus is in the environment, it's in decaying organic matter, and azole-resistant Aspergillus fumigatus infections has mortality rates up to 88% or even 100% in certain cases. Candida albicans is it is an organism that is part of our microbiota, but can become pathogenic in certain instances. So currently there are 4 main types of antifungals that are used in the clinic to treat these infections. And you will notice that it is much fewer than the number of different antibiotic classes that are available. So the standard first line of use is azoles, which inhibits C14-lanosterol demethylase. It's an enzyme in ergosterol biosynthesis. Polyenes bind to or they extract ergosterol from the fungal membrane. Echinocandins inhibit beta-1,3-glucan synthase, which forms a component of the fungal cell wall. And pyrimidines inhibit DNA RNA synthesis, and there's only one candidate, which is 5-fluorocytosine. So in addition, if my slide will go on, that would be nice. In addition, there are also 2 other types of antifungals that are not really clinically used that also target enzymes in the ergosterol biosynthesis pathway. So we see a very common theme here that ergosterol and its biosynthesis have been proven to be good targets for antifungal compounds. Unfortunately, there is currently drug resistance against all of these classes, and there are multi-drug resistant species and more are emerging. So this is a very big problem, and it's imperative that we develop new antifungal treatments. So what is ergosterol? So it is a fungal specific sterol. It is the fungal equivalent of cholesterol. So here you can see cholesterol and ergosterol. And it is responsible for maintaining fluidity of the cell membrane of the fungus. However, it's also an immunoactive molecule. It causes pyroptosis, and it is required for fungal pathogenesis. So there are many steps in the ergosterol biosynthesis pathway, and a large number of them remain unexplored as drug targets. So one of these is catalyzed by serol 24C-methyltransferase, or SMT, and this is the enzyme that we are studying. It converts cymosterol to ficosterol. It's a methyltransferase, so it accepts a methyl group from S-adenosylmethionine, and it transfers it to the cymosterol to produce ficosterol. So currently, this is very understudied. There is no structure available in the protein data bank. And this enzymatic step also is the step where cholesterol biosynthesis diverges from ergosterol biosynthesis. So there is no human homologue. So in the past, there has been gene disruption studies, the ERG6, which encodes the SMT protein. And researchers have seen that when they disrupt this gene, there are notable changes in cell physiology. The sterile composition of the cell membrane changes, and also the cell becomes more susceptible to metabolic inhibitors. So in 2020, Oliveira and co-workers did a study on Cryptococcus neoformans SMT loss of function mutants. So they knocked out the gene encoding this enzyme, and then they did tests. And very interestingly is that the organism cannot grow at 37 degrees Celsius anymore in either liquid or solid media. Then furthermore, they did a study on an invertebrate model organism, wax moths, where they infected these moths with the wild type or the loss of function mutant, and they saw that the loss of function mutant completely lost its virulence at 37 degrees Celsius. So there was no difference between the mutant and the negative control, while the wild type still killed the wax moths. So to move a bit on to drug discovery and design. So traditionally, drug discovery is done through high throughput screening, in which thousands and thousands of compounds are screened against the target. So this is done using robotics, and it's also very expensive, and it takes quite long. So the overall drug discovery process can easily take between 15 and 20 years, and it can cost between $1 and $2 billion. So this is a very expensive process. So what we want to do is we want to use the rational drug discovery method of structure-based drug discovery, and specifically using X-ray crystallographic fragment screening as a way to speed up this process. So just in a bit of more detail, for this, you need the purified protein, the target that you have chosen to investigate, and you need to get the structure by using a biophysical technique such as X-ray crystallography or NMR or cryo-EM, depending on which of these are suitable for your protein. So for us, we will be using X-ray crystallography. So after that, you will solve the three-dimensional structure from your experimental data, and then you want to identify fragments binding to your protein. So you soak fragments, maybe between 500 and 1,000 fragments, with your protein crystal, collect the diffraction data, and then see where in your protein these different fragments bound. And this is called X-ray crystallographic fragment screening. Then based on the binding of your fragments, these fragments are optimized and elaborated so they become larger until you can do biochemical evaluation. So this is often an iterative process where you modify your fragments, you co-crystallize them, and then optimize them further. And in the end, you will get a lead molecule that you can then take further through drug development. So what are fragments? So they are very small molecules, low molecular weight, and because they are so small, they bind with low affinity. So if you were to do a wet lab assay, you would have most likely missed this small compound because the readout would be so low because the binding is weak. But if you do a visualization technique, like seeing how the crystal binds the fragment, that gives you more information. So these fragments have to be elaborated either by linking, growing, or merging to make them into bigger molecules to have higher affinity. And like I said, this is an iterative process. So this brings me to the overall aim of the study, where we wish to end up, is to use X-ray crystallographic fragment screening to target sterile 24C methyltransferases from opportunistic pathogenic fungi to develop novel antifungal medicines. So these are the objectives. So first we have to produce the protein in an expression host. Then you have to purify it to high purity and homogeneity, crystallize the protein and solve the structure, and only then can you move on to fragment screening. So I will be focusing on the first three first three objectives, and you will see that unfortunately you can encounter quite a lot of experimental difficulty during this process. So the University of the Free State has an extensive yeast culture collection, and we have the type strain Candida albicans in this culture collection. So for the first step, we cultured this organism, we extracted the genomic DNA, we cloned it into an expression vector and introduced it into our expression host, E. coli. So this is a bacterium for the people who are not biochemists. And you insert the gene into the bacterium and it produces the protein for you. So after that, you evaluate if the protein has been produced by your organism. So the first thing we saw that we did not get any protein expression, protein production. We then tried alternative E. coli cell lines that are more susceptible or can produce toxic proteins. That also did not work. And then we thought that maybe this is an example of codon bias, where the codon usage between the two organisms are too different, and we therefore did a codon optimization of the gene sequence, and we bought the genes from Twist Bioscience, and we bought the genes from all 4 of the priority organisms in the WHO list. So then we tried again, we tried to express these proteins, and we were successful in expressing all 4 of them. So you can see there's a protein here at approximately 40 kilodalton, which is present in very high amounts. And after breaking the cells and centrifuging, we were very happy to see that the protein is still in the soluble fraction. However, before you can do any biophysical assays, you have to purify the protein. So you have to isolate it from the other host proteins, and it needs to be completely pure, as Prof. Kappa also mentioned in his talk. So these are the fractions that we collected after doing immobilized metal affinity chromatography, and we hope to see only one band here. And as you can see, the banding pattern for all 4 of these constructs are very similar, that you see this very pronounced bands at the bottom in all of the fractions. And that was very suspicious, because that is not something you usually see in with contaminating proteins in IMAC chromatography. So we thought that we will try anion exchange chromatography to see if we can get rid of these other smaller proteins, but we could not, telling us that they probably had the same ionic properties. And therefore, we thought that this can be a protein degradation, where our protein is being cleaved either in the cell or during purification, or it can be that there's incomplete translation, that the full length of the protein is not being made. We tried various things to solve this, to optimize our expression and purification conditions. We decreased the incubation time, we decreased the temperature, we added glycerol during purification, we had protease inhibitors as well, but nothing of this worked. We co-expressed with the molecular chaperone, and this is a protein that folds other proteins. So this improved the banding pattern, but it did not completely solve it. So this also told us that something about this protein is not folding properly. So what we decided to do then is to change gears completely and do construct redesign. So this was done in collaboration with our collaborators at the University of Oxford, and this was funded by the Africa Oxford Initiative that funded a research exchange for my MSc student who was working on this project. So the purpose of this is to now create a library of variants of the genes that are truncated at the N-terminus, and to put them in vectors that have either an N-terminal or C-terminal histag to see if we can get a boundary condition for stable protein production. So first, what we did is we made AlphaFold models of our 4 proteins, as there were no crystal structures or any other biophysical structures available. And we used a protein in the PDB with the highest sequence identity. So for example, for the Candida albicans SMT, this was a 4PNE, which was also a methyl transferase. So from the 4PNE structure, they observed that the N-terminus of this structure for the first 66 amino acids, there there was no electron density, meaning that the N-terminus is probably not folded properly. So these two were aligned, and we decided on different boundaries where we will remove amino acids. So for example, for the red part, the first 10 amino acids will not be there, and the rest of the protein will be produced. So doing this, the truncations were designed on a helix to helix basis, and for each of the constructs, between 6 and 7 versions of the protein were made. So luckily, we were quite successful in getting these variants to express. And these are all of the different variants that were made with their boundary positions indicated. So for example, for Candida albicans, we see that for the full-length protein and the first truncation, there was weak expression, but the further you truncate it, you saw better expression. The same was seen for the Aspergillus fumigatus, where we saw better expression for the these two variants, but if you truncate too far, the expression gets worse. Again, for Candida auris, the expression wasn't very great, and also not for Cryptococcus neoformans, so they will be optimized in future. So just to show you, this was the expression tests for the Candida albicans SMT variant. So the cells were lysed, and then a batch purification was done with nickel beads, and then an SCS batch was run. So here you can see for the N-terminal variants, numbers 4, 5, and 6 gave a better yield compared to the other ones, and these were the C-terminal HSTAC variants. So unfortunately, when the C-terminal HSTAC variants were purified, they precipitated. So moving on with the C-terminal HSTAC variants was not done. So for Candida albicans, we moved forward with these 3 variants and upscaled them for purification to do further studies. So just to give an idea about the workflow, so the cultures are upscaled, a larger volume is produced, they are purified by immobilized metal affinity chromatography, and then overnight the HSTAC is cleaved off with TEF protease. So this TEF protease is also a protease that is recombinantly produced with a HSTAC. So the cleaved HSTAC and the TEF will then bind to the column when you do a reverse IMAC, and only your protein will come out at the bottom. After that, we also did size exclusion chromatography to see the size distribution of the population. To do crystallization, the mosquito crystallization robot was used. So here you can see my MSc student Nokwanda working with Dr. Mike Fairfield at their laboratory in UK. And crystallization trials were set up with the APA protein, the methyl donor SAM, and the product that was left from SAM, SAH. So as you can see here, the purification was quite successful. And this reverse IMAC step was really necessary to go from a quite impure sample after IMAC to get a quite pure sample. However, after size exclusion chromatography, we saw that for variant 4, if you do not truncate enough, you still get a non-homogeneous population, which is a protein of 2 different sizes, and that is not suitable for biophysical studies. And if you truncate too much, you see the same thing again, that you get these double banding patterns where you have 2 different sized populations that make up your protein. Luckily for us, we had 1 variant out of the 6 that produced a homogeneous protein, and we could use that for crystallization. So unfortunately, no crystals were found with the APA protein. When the S-adenosylmethionine was added, the protein immediately precipitated, so that was also very unlucky. But with the S-adenosyl homocysteine, crystals grew between 4 and 7 days, but they were too small to use for diffraction studies. So follow-up screens were created, and we've managed to produce crystals that were the correct size. The diffraction data was collected at Beamline IO3 on the diamond light source in the UK. But once again, we were extremely unlucky that even though the crystals appeared very beautiful, they diffracted to a very low resolution of 5 to 7 angstrom, which is not usable for drug discovery efforts. So what we are busy with at the moment is we are optimizing the crystallization conditions. We have managed to recreate the purification of the protein in our home laboratory, and we have set up 96-well vapor diffusion screens with our RX Nano crystallization robot. And we used other sparse matrix screen kits that they did not use at Oxford. And we have also set up hanging drop vapor diffusion screenings to produce crystals that we can use for microscreening. We are also busy with assay development, where we are using the crude extract of the heterologously produced protein and purified enzymes to do biotransformation with the substrates. So just to conclude, we have managed to create an expression library of truncation methods for the 4 SMTs from the priority fungal pathogens. We have established a protocol for the purification of homogeneous Candida albicans SMT that does crystallize. The internal boundary of the construct, as well as the positioning of the tag, are both crucial to whether your protein is homogeneous and in a soluble form. And we could not crystallize without the addition of the acid-inyl homocysteine. So our crystals did diffract, so they are indeed protein crystals, which is good, but we need to further optimize the crystallization conditions. So the end of our goal is to do X-chem fragment screening experiments to identify fragments that then can be used to synthesize novel SMT inhibitors for the purpose of creating antifungal medicines. So I would just like to acknowledge the following people at the different institutions without whom this would not be possible, and also all the funders that have helped us to pay for this research. And thank you very much for listening.
Thank you very much for your talk. I now open the floor for questions. We have one question here on the floor.
Thank you very much for the nice talk. This is Marcus Newton, University of Southampton. It's very interesting and nice to have you at this conference talking on this particular topic. I have 2 questions for you. The first one is about a topic you mentioned at the very beginning of your talk. So you mentioned that pathogens or fungal pathogens are increasing globally. Could you comment on exactly why that is the case? And the second question is around your approach to handling fungi in general. So you're taking a protein approach to try and attack them, I believe. Could you comment on the possibility of using alternative means, perhaps a functional virus to deal with that? And by the way, I'm not a domain expert, I'm a material scientist, so forgive the lack of domain expertise. So please, if you could comment on those 2 points. Thank you.
Okay. So
Carmine, are you still
we are
online?
managing to keep more people alive, but yes, can you hear me?
Yes, you're breaking a bit, but we can hear you now.
All right, this question,
Oh, I think unfortunately we can't hear you. Can you perhaps maybe move a bit? Hello, Carmine, are you still online? I think there may have been a bit of a technical issue. Do you think is there something wrong with? It's not from our end. It's probably her end, right?
Yeah, she's online, but she's sharing and she's not muted, but
Yeah, it's probably the network. So if you can hear us, come in. Oh, she's dropped off. Oh, she's probably having network issues.
Sorry, are you still there?
Yes,
Can you hear?
sorry.
Yeah, everything
Yes, we
was
can hear you.
to my presentation. Of course, that's always how it works. I'm not sure. Do we still have time to answer the question, or are we out of time?
Yes, we do. Please. No,
All right.
yes, we do. Please, please start from the beginning.
So the one explanation that people say is that as the medical technology is advancing, we are managing to save lives of more people. But often they are immunocompromised also for a while at least. And this gives more opportunity to fungal infections. So that is the one thing. The second thing is they use similar compounds to, for example, the azoles in agricultural production because they spray the crops to prevent fungal contamination. So there you have this selecting for fungi that are more resistant to compounds that we use that are similar to antifungals. And then also with Candida auris, there's speculation that with global warming, this pathogen emerged and is more tolerant to conditions. So those are the speculations that people have on why we see this increase. It can also simply be that our monitoring techniques are more sophisticated, going more towards molecular biology monitoring techniques. So there are many things that come into play, but we definitely see more fungal infections and more resistant fungal infections. So that is quite concerning. Then other techniques. So in general, if you look at the current antifungals that are working, they are compounds that target specific enzymes or compounds that target ergosterol. One problem, though, is because the fungal cell is a eukaryotic cell, the difference between fungal cells and mammalian cells are not as big as between fungal cells and bacterial cells. So you do get much more toxicity from some of the antifungal compounds. So that is also why I think that targeting a protein that is specifically in the fungus and not in mammals is a good idea. So that, well, this is my knowledge, so I cannot really comment much on other techniques that can be used. So I don't know if that answered your question or not.
That's fine. Thank you very much.
Thank you. Any other questions from the floor? I see there's no other questions online, but I think since we got a dose of 2 questions in one from one speaker, I think we can call it quits here. Thanks you, Carmine, for sharing your talk with us.
And thank you for this opportunity.
Our next speaker is similar trend to what Carmine on drug discovery and protein crystallography. We have Ms. Lorato. She is here with us physically. She is from Wits University. Yes. I think you will need this. And oh, we need to have the ceremony of.
So, okay.
This is photography.
Official. Okay. Thank you.
Okay.
You can now pull this one down.
Yes, that okay.
Yeah. There we go. And then make sure you're sharing this.
You have to speak it to.
Okay. Now come back and you should be able to move. Does it work now?
Yes.
Awesome.
Good afternoon, ladies and gentlemen. My name is Lorato Mokoto from the University of Witwatersrand, and I'm here to present part of my PhD project, a very, very small portion, which is a mouthful nonetheless. My project is titled Supermolecular Synthesis of cis-1 amino 2 indinol derivatives, an in vitro and in silico analysis of drug efficacy against HIV-1 South African subtype C protease. So it's the 1980s, right? And we have a pandemic on our hands. The HIV virus is doing its thing. The HIV virus for 40 years has now been a public health concern. And with this, many scientists have actually dedicated their lives, time and effort, as well as resources in understanding the HIV virus. In order to do this, they've studied the virus in such a way that they have broken it down into different components and found that one target is actually one of the best targets to make drugs out of. So HIV protease is part of the replication cycle and is part of the last part of the replication cycle called the budding phase, where the virions actually leave the host cell in order to infect a new host cell. It is important because it cuts the polyprotein into functional units, which infect the host cell. And mutations of the protease actually affect the mutations of the protease actually help affect the efficacy of drugs and is seen in drug resistance. So what I wanted to do, I wanted to create drugs or synthesize and design drugs that are able to be potent and favorable pharmacologically and present good bioavailability, tissue penetration, as well as minimal side effects. To do this, I started off by actually trying to modify the current protease inhibitors. I started off by trying to modify the protease inhibitor indiniver by using a molecular synthesis technique called one-pot synthesis, where I reacted the indiniver itself with 1-propanol for 4 hours under these conditions. And then we found that a crystal formed. But there was no modification of the indiniver itself. Instead, indiniver cleaved into 2 sections or 2 portions. 1, lactone, which remained in the solution, and 2, the crystallized portion, which was the bis cis-1 amino 2 indinol crystal. So this crystal, when analyzed or elucidated using X-ray crystal, single crystal X-ray diffraction, we found that it formed into the monoclinic crystal system and space group of P21. The crystal was then affectionately named MLM000, which formed as a blueprint for the next derivatives which I was able to synthesize. So using cis-1 amino 2 indinol as a blueprint, I then synthesized a couple of derivatives, 6 derivatives in total. However, I will be presenting only 2 of the 6 derivatives as well as their efficacies. So to do this, I continued by using the one-pot synthesis method and reacted the cis-1 amino 2 with a solvent, a aldehyde, under acidic conditions to form nitrogen analogs. From there, we elucidated and characterized these structures using FTIR, Raman, as well as single crystal XRD. From the Raman spectroscopy, we analyzed that, yes, every compound that we did, the Raman and FTIR, we analyzed that, yes, every compound that we did, that we were able to synthesize was actually very different from the starting material itself. So there are functional groups that were added and functional groups that we were able to characterize in order for us to build these structures. As I had mentioned that in this presentation, I will only be mentioning 2 of the 6 that I synthesized. And we also did single crystal XRD on these compounds and found that both of them are within the orthorhombic space group in the orthorhombic crystal system underneath the space group of P2121. So in order for us, we have these crystals, that's great, but now we need to test their efficacy. We did computational studies using molecular docking and simulation studies by first docking the the compounds to the protein, and then simulating what the possible interactions between the protein and the ligands would be. As we see here with MLM000, the ligands are very dynamic, meaning that the traditional way to do drug design is to actually have a compound that would interact or dock as well to an active site, which is sitting there, and then stay there, and it would be considered a tight binder, meaning that it would actually compete with the substrate, the natural substrate, and prevent proteolytic activity from happening. However, with my drugs, we saw that they were actually quite dynamic, meaning that they had a high affinity for the whole protein instead of one portion of the protein. So with MLM, we see that because the compound is held together by hydrogen bonds, it actually breaks apart, and each portion of the drug actually has high affinity for the entire protein instead of just one portion of the protein. So we did molecular dynamics and docking as well as on the other compounds, and we saw that the bulkier compounds actually had an affinity for the active site and just stayed within the active site, whereas the smaller compounds, just like the MLM000, had a high affinity for the entire protein. So this could work to our advantage or could work against us. But we saw in the post-dynamic analysis where MLM000, 035, and 034 were actually baselined against the ApO structure. And we saw that the red one, which is 000, and the yellow one, which is 035, the smaller compounds there, they actually do destabilize the protein, which can put the proteolytic or catalytic efficiency of the protein into question. However, the more bulkier compound actually made a more rigid protein, which could also work with us or against us. We also did an RMSF value evaluation where we saw that various portions of the protein were actually affected by the presence of the ligands. The ApO structure, which is seen in black, was baselined against the structures which were analyzed with the compounds with them. So we see that in the active site, in the fulcrum, the hinge, the flaps, and the cantilever, there is some form of a destabilization where 000 and 035 now exhibit a more flexible protein, and 034, which is seen in purple, actually exhibit a more rigid and compact protein. Great. So now we did the computational work, and we wanted to see, will these compounds that are synthesized actually have any activity towards the protein itself using empirical studies? So in order to do this, we used a PET11B plasmid, which was transformed into BL21DE3 cells, and then protein production took place where the cells were lysed, dialyzed, and then purified, and then analysis of protein purification took place. After acquiring our protein, we then performed an enzyme assay, and then from there, we were able to determine enzymatic parameters to see if our protein is actually healthy. And in order to do these, we actually, from the parameters that we got, we baselined them against literature that was found, and we saw that, okay, cool, our protein is actually healthy and it is working efficiently with a good turnover, with a good catalytic turnover. From there, we then moved over to inhibition studies. So with the inhibition studies, we first did an IC50 evaluation. From doing the IC50 evaluation, we then used the Chang-Persoff equation in order for us to obtain the inhibition constant. We saw that compared to Indinavir, our compound, MLM000, the more smaller ones in comparison to the more bulkier one, and 035 actually had better inhibition constant in comparison to MLM034, which had a higher inhibition constant, higher than any other protease that is even on the market. We also did a cytotoxicity evaluation, and because it is known that Indinavir is actually toxic towards kidney cells, We did a cytotoxic evaluation against various cells. And with that, we saw that, yes, indinavir is indeed toxic by looking at the LC50 obtained. However, the drugs that I was able to synthesize are less toxic. We also did a cell viability evaluation where we used untreated cells as a baseline in order for us to determine how many of the cells are actually still alive or how many are dead after treatment with the respective ligands. And we saw that indinavir actually had the highest amount of cells that are dead in comparison to any of the other drugs. We also did a evaluation against DMSO because all of the drugs were actually reconstituted in DMSO. So we wanted to make sure that whatever result that we did obtain was actually from the drug and not from the effects of the DMSO. Okay, there we go. So in conclusion, I was able to synthesize drugs that do not violate Lipinski's principle of 5. They are all less than 500 grams per mole. And these drugs computationally actually exhibit moderate binding. However, they also exhibit dynamic or transient behavior. This could work with us or this could work against us. Actually, this could work with us or this could work against us. We also saw that the drug molecules actually exhibit enzymatic inhibition, which is good. And considering that these drugs are less toxic, they could actually be considered for pharmaceutical use. I would like to thank my mom. I would like to acknowledge my mom, my supervisors, Professor Yasin Syed and Dr. Mark Smith, my mentors, and as well as the Protein Structure Function Unit, the National Research Foundation, and the National Research Foundation. Thank you.
Thank you, Lorato, for your exciting talk. Is there any questions on the talk? Yes, Philip at the back.
All right, nice presentation. Though I'm not in your field, but what I want to ask is just something. Yeah, you mentioned that the drug molecules are less toxic and a bit better than the approved one. From in your findings.
Yes.
Okay, what I'm saying is, I'm not in your field, so if I ask, maybe just have to bear with me. But you, based on your findings, you have said the drug molecules are less toxic.
Yes.
And then a bit better than the approved one.
Yes.
So my question is, what about in terms of the cost? What can you say about that?
In terms of cost?
Yeah.
Considering that the drug molecules
into
which one?
Oh, yeah.
Can I use this one? Okay, sorry. Considering the chemicals that we use or reagents that we used, those are basic reagents that you can actually find in the lab, which are less costly. And the process to actually synthesize and time that it takes to synthesize is way less in comparison to actually build all these bulky molecules. So it's basically a tiny little molecule, which takes about 24 hours to make and then 2 weeks to crystallize, compared to what we currently have, which takes months and months and months to actually start building on various functional groups and various analogs onto these things. And yeah, it takes way less time and it is less costly.
Okay, I think with respect to time, because we're a bit slow and we're eating into lunch, not yet, but we have one more speaker after here. Any quick questions? But if there isn't, and nothing from online, no. Let's thank Lorato for a talk. Our last speaker for the session is Gideon from Wits University as well. Gideon's presentation were much similar to Kudo's one on imaging. This is not a mic. You have to just pick up.
Okay. Can you hear me now? Okay, thank you very much. My presentation is going to be on X-ray phase contrast imaging as an alternative to laboratory-based X-ray sources. So I'm going to talk about the overview of conventional methods, as well as the motivation to this work and what we want to achieve, as well as the methodology and some of the results that we have so far, and then what we are planning to do in the future with the work. Okay, just a brief overview of what we spoke about in the lab yesterday. This is a schematic of what we normally get in a lab-based X-ray source. We've got an X-ray source, a rotating stage where we put the sample on, and we have the X-ray detector, and then we use a cone beam computed tomography system. And in this system, what you have is that according to the Lambert-Beer law, there's a reduction in intensity when X-rays pass through objects, and which is given by that formula where I naught is the initial intensity of the X-rays, and then you've got the attenuation coefficient, and also I being the intensity of the X-rays that reaches the detector after absorption. So normally, traditionally, what we get in a lab is absorption-based imaging. And then in 2022, I was an intern due to the LEM grant at the ESRIF, and we worked on the BM05 with Dr. Kudakwashi Jakarta. That's one of the samples that I actually took there. It's a crocodile from Zimbabwe. And by using phase contrast computed tomography methods, I think the question was then, whilst we wait for a synchrotron in Africa, what problems can we solve, or what additional methods or improvements can we do in a local laboratory to solve this issue of contrast? Because of most of the specimens that we work with are fossils. And usually there's an issue with absorption contrast when there's a similarity in contrast between the rock matrix and the fossil, the rock matrix in which the fossil is embedded, and the fossil itself. So to try and solve that problem, at least for smaller specimens, the question was, how can we improve our lab? And then that was when the thought of introducing what is called a phase contrast. At this stage, we have only done X-ray imaging, but we are hoping to improve this to do phase contrast tomography, as I'll explain later. So we purchased this Taubert low interferometer from Germany. As you can see, it has got 3 gratings, the G0, the G1, and the G2, which I'll explain shortly how they function. You also have a piezoelectric motor and the connecting cables, as well as the breadboard that is mounted on. I think the main issue in a lab-based source is that we are having a polychromatic X-ray source. And that being said, one of the needs of us being able to cultivate this phase contrast imaging is that we need a coherent beam, because we need interference to occur. And one of the requirements for interference in such cases is coherence. So there is the X-ray source. If you remember the image from yesterday, I think I showed you the X-ray source. And then between the X-ray source and the detector, it's where now this Tallinn system, which is an Taubert law interferometer, sits. So you've got that G0 grating. Because we are producing a polychromatic X-ray source, you need to create some form of spatial coherence in the data. So that G0 grating is an absorption grating with which has gold bars, and then it has some slits in between. So what happens is that when the beam exits the X-ray source, the part of the beam that passes through the gold, because it's highly absorbing, it's actually absorbed at the ideal working conditions, which is about 40 kV according to the design of this instrument. Then slits between the G0 grating now act like single point sources for X-rays, allowing a more coherent beam to actually pass through, which is needed for interference to happen later. So it's like, and then of the slits is highly dependent as well on the period of this grating, which is needed for this work. Then the sample is placed just before G1. So when the X-rays pass through the sample, there is absorption, there is diffraction, and there's also a scattering that is caused by microstructures within the sample that occurs. And then you have the phase grating. And then after that, because our detector is initially made to read intensity, it reads intensity values. So the function of the G2 grating is now to take those phase variations into a more readable value, which is intensity variations that would then be projected. That's basically how this Talbot-Low interferometer system works. So what we do is when you put the gratings at the distances that you need to do to have for you to get an interference pattern, what we first observe are more fringes, which are as a result of superposition of these gratings. And then you find out that there's a periodic pattern that is produced or generated by the phase grating. And then the analyzer grating itself is also has also its own periodic structure. And what you do is you find out if there is a superposition of these gratings, and then there's a slight misalignment, you get those more fringes. And then there is a quantity that we use to measure the image quality in such a system, which is called the visibility of these more fringes. And according to that formula, Imax minus Imin gives you the amplitude, and then Imax plus Imin over 2 gives you the mean intensity. So it's just the ratio of the amplitude to the mean intensity, which is why I've just multiplied by 2, because it's Imax plus Imin over 2. So this visibility is more or less an image quality that we use in this imaging system. So having said that, the way we do this phase contrast imaging is that we perform what is called phase stepping. So the grating that we're using has a period of about 6 microns. So across the 6 micron period of the grating, you need to have steps in between that you acquire the data, and then across the whole period, and also getting a full wavelength of 2 pi. So what happens initially is you have an X-ray beam that is coming, and when the X-ray beam comes, you perform what is called phase stepping. In this phase stepping, what happens is that initially you've got the slits covering all the bright fringes on the detector on the alignment. So what you get is a 0 value in terms of intensity on the detector. And then you start moving your grating across as you do the stepping. So for example, in most of the experiments that we do, across the 6 micron period, we use about 20 steps, and we just overlap even one period just to actually get maybe a period and a half. So we use steps of about 0.4 microns. So during the phase stepping process, like I said, you initially have your gratings covering all the bright fringes. So the value that you get on the detector is 0. Then as you step across, you see that the intensity values continue until they reach a maximum, whereby your grating is now covering all the dark fringes and only the bright fringes are appearing. You have a maximum value on the detector, right? And then the process continues, you have a negative maximum, and then you get also a full wavelength over the period of the grating. And when you place the sample now in the path of the beam, there's going to be there's going to be refraction, there's going to be scattering, there's also going to be absorption of the X-rays, which in turn distorts the initial path of the beam. And then after doing that, you then compare, that's how the image is then produced. You get first stepping caps of each pixel that are compared, and then you obtain the results from these alterations caused by attenuation, like I was saying, the refraction and the scattering as well. And then from these, we can then get the absorption, the dark field, and the phase contrast images, which is calculated usually by a software. So here I'm just again showing what actually happens there. You've got showing what actually absorption happens. You've got absorption of the beam, so there's a reduction in the amplitude. There you've got a phase shift when the sample is placed in the path of the beam due to refraction. And also when there's scattering, you also find out that there's a reduction in the mean intensity as well as the amplitude. So the black lines reflect the phase stepping that happens for a sample is put in the path of the beam. And then those altercations from these intensity values are then the ones that are used to calculate the results that we actually get from this data. So like I said, one of the image quality metrics that we use is the visibility. And in our system so far, the maximum visibility we're getting is at about 0.30, which is about 30%. So there I'm just showing the visibility of the reference, whereby you are having the phase stepping as it occurs over the number of steps. And this time I used about 2 periods, which was about 8 microns. I used 2 periods, which was about 8 microns. And then there's just the feed showing how a perfect phase stepping curve would have been if we obtain a perfect visibility reference image that we use for this data. And then, like I said, that at the ideal AV, we expect that all the X-rays that pass through the gold absorption gratings are absorbed, such that we get some form of highly spatial coherent beam. But then these gratings are desired to function at about 40 kilovolts in a lab. And you find out that most of the specimens that we work with require more than that. So now you start to have a trade-off between visibility and actually visibility and then the quality of the image. Because now what happens is as you start increasing your kilo electron volts, so the interference occurs at Talbot distances, which is so the Talbot effect is self-imaging phenomenon that happens where self-imaging structure like a grating produces images of itself at different distances across. So this formula is given by 2p squared over lambda, where p is the period of the grating and lambda is the wavelength. So what happens is now as you increase the energy, now because we are operating at a Talbot order of 1, it means that we cannot adjust any more parameters. So we start to have some form of transmission within those gold cut gratings because they are designed to absorb all the energy at 40. And then that means that our coherence beam starts to get affected. Then the image quality decreases. And as I said, because the beam becomes less and less coherent. And this is an experiment that we did to actually see, okay, at what stage can we increase our KV to in the lab and still get meaningful results with meaningful visibility, yet also achieving the results of getting a desirable image. So one other experiment that you find is that in any mechanical system, what you find is that you get sometimes friction and internal resistance that occurs. So like I said, when we do the phase stepping, we've got a piezoelectric motor that is connected to the G0 grating. So that's the one that pushes the grating across. And this experiment was just to see how well can we reproduce our results so that if we see any variations, we know that it's due to the sample's properties, not fluctuations in the system. So what we observed is at the point of contact between the piezoelectric motor and the G0 grating, when we repeated the experiment, there was more of like some variations in between. So what we did was then to drive the piezoelectric motor so that there's enough contact and we deal with this hysteresis effect, and then to a point of about 0.5 millimeters inwards, and then we define that as our absolute zero. Now, to say that we can start here, and immediately after the second experiment, there was some form of repeatability showing that it's essential for us to get repeatable results to just play around with this hysteresis effect and our reference point within the experiments. And one other thing that we also did, because we have this grating, so the whole distance between the G0 grating and the G2 grating is about 60 centimeters. And as it happens like that, G0 grating, for us to get an interference pattern, is placed about 7.5 centimeters from the X-ray source. So what this entails is that at the point where we do imaging, somehow our resolution decreases. So the other question was, at what point again can we trade off resolution for sensitivity? Because we want our imaging system to be able to detect any small variations in the phase shift. However, we also want to be able to get enough resolution in the data. So we did this experiment whereby I used a marker and just placed the different distances from the G1, which is the phase grating, and tried to get different results. As you can see that as the sample moves closer to G1, we obviously get a sharper image and an enhanced image, and as also move further away, the sensitivity becomes low. But again, there's a trade-off, and we also need to come up with as to how far further away from the G1 can we go, but still getting enough results. And just to show this, we did a contrast analysis whereby you see that the red line depicts 18 centimeters away from the G1, which means the sample was as close to the source as possible, which means the resolution was high. And as you can see, there is a low contrast due to the distance between the peaks. And the further away we moved from G0 towards the phase grating, there's higher sensitivity, there's more contrast. And again, you can see there's a third peak showing that there's probably an overlap that happened between the previous two images in terms of contrast signal. So it would be ideal to have your specimen as close to the phase grating as possible to get high sensitivity. But however, with some specimens, you just need to find an ideal point where you still get, like I said, the trade-off between resolution and sensitivity of this data. And then just to show proof of concept, we used the first experiment, successful experiment rather, was the circuit board that I just found. It's I think it's a RAM chip actually from one of my old laptops, and then just did an experiment to show how the three contrast signals appear in the data. There isn't really much to conclude from this, but you can see that there is actually enough to show how the differences between these three contrast signals. And part of the reasons why I showed you the beetle in the lab yesterday, it's because it's one of the specimens that I'm also trying to use to do tomography with in the future. And as of now, we've just done X-ray imaging with it. And as you can see, the first image depicts how the image appears in an absorption contrast. The second one that's actually dark field imaging, and the third one would be the phase contrast. And you can see the images are quite, they show different contrast between them. So we'll try and use all these three methods to complement one another. And to describe this, because we are not biologists, we'll partner with people that study these various specimens and see what research questions they have. We apply the technique and then we collaborate together just to show how our system can solve a local problem within the laboratory. So this is what we have so far. Then, like I said, the plan now is to move and do tomography. So as you can see, initially when I showed you the first image, the grating was sitting on the rotating table. That means we could only achieve to do X-ray imaging. But with the help of my supervisors, they managed to design that arm, which now allows us to tweak the gratings upside down so that to allow the sample to rotate, and then we can move and then do X-ray phase contrast tomography in a laboratory for those small specimens. I would like to thank my supervisors and financial support from past. And lastly, I saw on Tuesday when I was chairing that Philip shared some of his pictures from the LAMP experience in the laboratory. So I also wanted to break that because of LAMP, I managed to go to France, and for the first time, I did some skiing. I went to PSG, I went to the tower. So it was a very good experience. And again, we had to export some of the knowledge that we got during that experience to the lab. Thank you very much.
Thank you very much, Gideon, for your exciting talk and the nice pictures from France. I'd like to open to the floor. Any questions? Yes.
Thank you for the nice presentation. My question might be very basic. So I wanted to ask about the gold gratings. You said at around 40 kV, that's like the limit for them. So I wanted to know if it's because of maybe the absorption properties of the gold material, or is it the design of the grating material?
So according to the manufacturer, at 40 kV, because remember I said that you've got the gratings, the absorption gratings designed such that there is gold, and then there are transparent slits in between. So at 40 kV, it's like the point where we get maximum absorption of the beam that comes through those gold cut gratings. And the only transmitted beam theoretically is within these slits, because we need a coherent beam. That means that these slits now act like point sources for creating a beam that will interfere in the so at 40 kV. Basically, the answer is that that's where we get maximum absorption through the gold gratings, and that means where we get maximum image quality in the data.
So the beam that is transmitted through the gratings,
the slits, yes.
interferes constructively
and destructively. That's why we get bright and dark fringes on the detector. So it's like constructive and destructive interference.
Oh, okay. Thank you. Thank you.
Yes.
Yeah, I also want to thank you for your talk and congratulate you, Gideon. And I'm very happy we got someone like you developing this expertise in Africa. So I always had expected the phase contrast imaging to have a much greater sensitivity. Basically, the number of 100 to 1000 is bandied around because of the phase contrast image coming from the real part of the refractive index and the absorption contrast from the imaginary, and looking in the area of X-rays, you know, what's the relative sizes of these components. But then I was a little disappointed to see both in the beetle and the memory chip, the phase contrast didn't look as clear as the absorption contrast. So I was just wondering if you could comment on that. Will it start to perform better as you progress in the research, or is that what you can see, or do you have to look at the tomographic reconstruction before you really see the improvement?
Okay, I think first of all, I'll comment on the choice of specimens. These were just random specimens that I was picking through to see how they perform. But the plan now is to try and maybe study biological specimens more that less absorbing, more transparent to X-rays, but have got a higher refractive index, maybe, and start there. So like I said, most of the work that I presented here is characterization. So we're still trying to come with an optimal type of method. So yes, to answer your question, it will get better. I think also we need to look at the type of specimens that actually exhibit maybe phase variations better than absorption. Or for example, if you're looking at biological specimens, we're looking at the dark field imaging, whereby there are microstructures causing this X-ray scattering, so we can see a better dark field image. So I think it's just playing around with the parameters, yes, but also looking at the right kind of specimens and coming up with a protocol to say, okay, if this system, but for now, we can work with 1, 2, and 3.
And can I follow up?
Yes.
So also, what's interesting me is the image. What are the units on the Z scale, if I can put it that way, with the phase contrast? Because the idea is the phase contrast proportional to electron density.
Mm-hmm.
Do you finally give a result in electron density, or is it in some arbitrary unit proportional to electron density? Or how would you calibrate it to actually be electron density?
So you mean the units on the
color scale, the
on
Z
the,
scale,
on the color scale,
the gray scale?
Okay, so that was in terms of the phase change. It's actually like using the d sine theta is equal to n lambda. It was actually in terms of phase change.
Yes, but I've seen that is directly proportional to electron density. So one would imagine you could calibrate it to that.
Okay, I think that's something to look at. Not yet done.
Okay.
Yeah, thank you very much.
Any further questions to Gideon? Any questions from online? No questions from online. Well, if there's no further questions, let's thank Gideon. According to the program now, it is lunchtime. So thanks for your participation in this session. Any notices? No? Nope. So lunchtime. Thank you.